So I want to thank maybe you want to put up your slides, Ranja. I want to thank everyone for being here for our AI and biocuration workshop. We're very lucky to have started this, I don't know, maybe 6 months ago, when a whole series of discussions were starting about how different folks are integrating AI into their actual workflows and how they're not just using it, but how they're responding to a sort of rapidly changing environment and setup. Almost all of the setup work for today's workshop has been done by Ranja. And this is my very brief introduction. I'm going to hand it right over to Ranja to introduce our speakers and to kick us off so things are actually moving forward. And thank you all for being here. And, be sure to sign up and join the society, and we'll be having more of these. And so that's a really fair way to follow on and keep being involved. So thank you all for being here. Ranja, it's yours.
Thanks, Mark. Yeah, I'd like to welcome everybody as well to our second session of the AI and Biocuration virtual series. So today we have three great talks, and I'm very excited to introduce the speakers. Our first speaker is Lisa Mathews from Reactome. So Lisa earned her doctorate in genetics from Cornell University and has more than 25 years of experience in biomedical knowledge representation, proteomics databases, pathway curation, and scientific data integration. She previously worked on quality control and annotation pipelines for large-scale proteomics databases at Insight Genomics, Proteome, and has served for the past 17 years as managing editor for Reactome at NYU Grossman School of Medicine, where she oversees pathway curation workflows, coordinates communications with subject matter experts and collaborators, and manages quality control processes and quarterly database releases. So before Lisa starts, I just want to say that these talks will be about 15 to 20 minutes. Maybe we'll take one or two questions right after the talk, but remember that we have almost like a 25 to 30-minute open session at the end where people can put questions in their chat, and Mark will be monitoring the questions, and we'll get to as many questions as possible. So with that, Lisa, take it away.
Okay, thank you. And thank you for the opportunity to talk to you today about the progress that we're making right now, working with LLMs and AI agents to try to help curators in the process of pathway curation while still maintaining the high quality of annotations. So turning things over completely to AI is definitely out of the question, but we want to see if we can come up with ways of speeding up the jobs and tasks the curators have. So as most of you know, Reactome is the largest pathway knowledge base for manually curated open source pathway data. And to date, we have over 2,800 pathways curated from over 42,000 literature references. But as all biocurators know, the rate at publication, biomedical publication production is vastly out exceeds the rate that we can actually integrate this information. So that's really the impetus for trying to get some sort of automation in our pipelines. So we started by looking at now whether AI agents, more sophisticated agents like Claude, can be trained as pathway curation assistants. And to understand our approach to this, I'm just going to walk really briefly through how curators annotate pathways in Reactome. So generally, they start from review articles from the literature. They assemble their own literature base based on their understanding of a pathway. Sorry. And then they
Lisa, do
create a
you want to
framework.
share your slides?
Oh, I'm not sharing this. Oh, gosh. Sorry. Did not realize I was not sharing. Why is this not working? Okay, can you see it now?
We can. It's great.
Okay. Yeah, so we're just trying to decide how we can use Claude now in automated curation. So the process that curators use for annotation is basically taking review articles from the literature and creating a framework for a pathway or topic of interest, in this case, the immune system. They break that down into subpathways and then the component reactions within a subpathway. And then for each reaction, they collect a lot of information, including the biomolecules involved, the reaction, the proteins, chemicals, sets, and RNAs and DNA molecules. They cross-reference these to external databases for interoperability of this resource with others. And then we also annotate molecular functions to catalysts, and we identify a location for all of the molecules within the reactions. Where possible, if we're annotating diseases, we cross-reference disease ontology. And importantly, for all reactions that we are that we put in the database, we have direct experimental evidence supporting them. So with this information that's collected, curators enter it via the curator tool into the database, and this information is then used to create a manually drawn pathway annotation, pathway diagram. And for the entire pathway, then these are assembled to inform the creation of a textbook style interactive diagram with which the users interact when they're looking for content in Reactome. So how do we automate this complex process? The curator guide, I mean, training curators is a very long process. Most of us have been at this for more than 10 years. The curator guide is over 400 pages. There are 200 classes to keep track of, and this is all integrated into glossaries. So the question is, how do we get a handle on all of this? And now that Claude and AI agents are able to manage large amounts of information, I sort of took a simplistic approach and saying, well, if we provide Claude Pro, for example, with all this information, and then design a prompt that explains what we want it to do, can it create the framework for pathway annotations, which curators would review, obviously, and verify before we would integrate them into the database? So this is a simple question that we asked. Here are these guides that we have. Please create this pathway annotations for this pathway, which I chose, which is a pathway we had not yet annotated. And almost, within less than a minute, it was able to tell me that it was going to be able to create a structured curation record that included all of the important information that a curator would normally use to create that reaction annotation, for example. Then it created a framework for the pathway, proposing the subpathways and component reactions within it. And interestingly, it was catching already the post-translational modification. So down to a very detailed level of information that we generally capture. So this was encouraging. I did have some back and forth with Claude because one of the traps that we fell into when we were analyzing ChatGPT's ability to annotate pathways was that it was very hard to track down the literature references that it was citing, was fabricating what it was presenting as evidence. So to avoid wasting time, I asked any time that it was presenting information or references to external databases to provide a link so that a curator could easily click on it and get to that information. So it was actually able to create a pretty reasonable table style output in which it did include the links. The links were accurate. And then we had curators review the actual annotations for the total 18 reactions that were proposed. And globally, it got most of the information correct. There was information that was missing on occasion. It would miss go terms or post-translation modifications. When it created a summary of the reaction, it was sometimes providing sort of unfounded statements. And a few of the literature references were not actually correct. So we decided to see if there were ways to improve the performance of Claude Pro by providing it access to a better prompt, more sophisticated engineering of the prompt. So mine was very simplistic. And then just provide access, MCP server access to Reactome and also to OLS, because a lot of, as I said, we cross-reference to a lot of databases that are within the OLS. So we set this up. So the improvements that we expected, so the system prompt adjustments really focused on verifying all references that were, rechecking. So you find a reference when you're going to spit it out to the curator, please, verify that that is right. So we'll check, for example, matching names and title keywords and things like that. And and then also, in addition to that, to make it faster for curators to actually identify the places of interest in the papers, I asked it to extract the text excerpts as supporting evidence. So the goal here was to improve the references, to automate the ontology lookup so that that was correct and complete. And also, it gave an opportunity for us, because curators, when they're generally annotating a pathway, do a normal pathway, and then later they might come back and do disease annotations. But here I thought, well, okay, if we have access to Mondo and Do, the disease ontology, if there are proteins involved in the reaction that have been implicated in a disease state, then point that out to the curator as well. And so providing access to the Reactome MCP server provides the benefit of being able to cross-check the annotations that it's creating to look for example, duplication or other connections to pathways that maybe we hadn't considered. So, so in the first sort of pass of testing this, we did find this this improvement of the prompt did fix all of the references looked at so far in terms of their correctness. And the primary evidence was being extracted, and that was proving useful to guide the curator back to the place in the article that they should look at. And so looking at the annotations coming out, this is again in progress. So I don't have a lot to share, but generally the inputs and outputs, the general characteristics of the reactions are correct. It's actually pointing out some of its own inconsistencies. So compartment here was sort of flagged where there were some inconsistencies in what was stated in the references. So that pointed the curator to where they needed to focus on maybe some inconsistent data. And then also we found that it was proposing some unexpected suggestions, and that was based on the fact that the curator guide that we had provided it had a sort of misinformation. So that actually helped us correct some of our documentation. So just again, a really preliminary report on this. So the first pass of this took about 4 hours to generate the reports, discussions back and forth with Claude, and then another 4 hours to check. This, I think, will come down the amount of time that it takes as we learn to engineer the prompts a little bit better and yeah, just tweak the process. And the checking may or may will probably come down a little bit as well. So, but globally, what we're finding are the pathways themselves are accurately outlined. They the core reaction attributes are faithfully captured, at least in the cases that we've looked at. We haven't looked at the most complex types of annotations yet, but it's getting, the fact that it's post-translational modifications correct is pretty encouraging, I think. And the evidence extracted is supporting the assertions that it's making, so that was also encouraging. One of the limitations of this is that obviously it's not free to use this. This is Claude Pro. I got my own subscription to it because I was inspired by Chris. Really, I wouldn't be here today talking about this if I hadn't listened to Chris's talk 2 months ago. And I think so it is it's a cost, but I think what it is able to do is encouraging, and I think that we've decided in the group that we will actually get subscriptions for all of the curators, and for the next month we'll be testing this in different ways. So again, we'll test more topics and get feedback from the rest of the group. I'll just touch on a couple of other things that we have experimented with. In addition to just annotating a topic or a review article, we're also looking at one of the bottlenecks in curation is really getting data into the database, and less so, identifying papers and reading them. But it's the data model is complex. There's lots of information to go in. So we wondered whether or not we could take certain types of curator notes and convert those into annotations directly or with some help. So this is a very simple case where we're annotating the cell mark. So in a cell lineage path, as cells progress from one cell type to another, they express different markers, protein and RNA markers. And so a curator captures all the information about the markers in one cell type and the references for those markers. And in this case, the curator tabulated it. And in this particular case, they had already entered the information into the database, but I was interested to see what Claude was able to do with this, if we explained what we were doing and then asked it to sort of convert that. And so here, this is just showing the results that Claude came up with, the annotations, and here I've compared them to what's actually present in the database, what the curator entered. And the interesting thing that we found out is in all cases, the annotation that Claude proposed that wasn't in the database was either missed. It's something that the curator missed from their own table or hadn't yet entered, or in two cases, the issue was that there was only one literature reference supporting that, and that's a criteria. It has to have at least two literature references. So it was something that was missed that wasn't explained in the prompt. So I think that's also encouraging that we can, take formatted curator notes and look at integrating those as a way of speeding things up. So, and then just a few other things we've been doing. Documentation, obviously, now that we have access to the MCP server, it's just a window into the database that we didn't have before. So normally we needed developers to help us write queries to query the database, but now, we can ask Claude to take a look at, here's our data model glossary, take a look at, the schema. Are we missing anything? And so we've done that. And then Curator Guide, obviously, I pointed out places where it's showing us mistakes in our own description of processes. So that will hopefully be another area where we can improve and update our documentation with a lot less effort on the curator part. QA again, this is game changing for me since I have to handle a lot of the QA. Now we can actually, because I'm not a Cypher expert, and we're going between SQL and graph databases now. So I've really relied on developers to create those scripts for me. But now in plain language, you can ask Claude, describe the class and what you're looking for in terms of quality control, and it writes the Cypher query for you. And I've tested that in at least one case, and that's proven to be effective and useful. The last thing is internal review of Reactome pathways. That's another chore that curators take on. Once another curator's finished a module, we pass it internally to make sure that the content passes quality checks. And now Marc has actually spearheaded the development of a pipeline that looks at the pathway reports that the curator generates before we send them out to external review. And so that has had favorable feedback from curators so far. So as I said, we're going to be spending the next month testing this more on larger scale. And so we've created a GitHub repository where curators are creating their own skills and entering them here so other curators can make use of them and make improvements. And so we're really just now getting started in the last couple of weeks, so I don't have much to say on that, but hopefully more to say in the future. So just wanted to say again, thank you to, I'm really grateful for this forum because I really would not have thought to get started on this if Chris Mungle hadn't said, oh, curators should really be doing this every day. I'm thinking, well, I don't do it every day, so maybe we should try. So I think it's really paying off for um, use in Reactome. There's a lot to do and a lot to test. I sort of painted a rosy picture, but hopefully I'll come back and maybe give an update with more substantial data. So I just like to thank everybody in the group that's been involved in helping develop this or talking about it. And yes, thank you again for letting me present this.
Great. That was very exciting to know that Claude is working for you. Do we have any questions? People can either, raise their hand or type in the chat and we can read the questions. Victoria, you can unmute yourself.
Hi, Lisa. Thank you so much for the talk. I gotta say, I feel the same way. I think we probably attended the same Chris talk. I was also inspired. I also trying to do these things with Claude. Probably I don't have the same resources as Reactome, but I'm trying. So I've been trying to do the same things with similar results, I think, to you, which is encouraging because it seems like we're on the good path, probably. And I wanted to ask you something, might be a bit silly, but you also mentioned ChatGPT. So I think you probably compared the two of them. I'm trying to push for Claude in my group.
Yeah, yeah.
So I wanted to know your feedback on ChatGPT.
Yeah. That's a very good question, and it's something that we should do. The problem, so I started out actually really wanting to quickly jump into the MCP server, the ability to integrate that into the pipeline. And I could not find out. Claude made it so easy. I did that all on my own. I didn't have to ask anybody. I set it up and with chat, it was just much harder to see if or how that could be done. So to be fair, I mean, to be clear, I haven't really tested that just because I can't do everything I can do with Claude, but we will. Other curators are looking at different tools. Some are using Google Workbench for some processes, and I don't know that anyone. Mark, do you know if anyone is using? I am not aware of anyone using chat right now, just because of the limitations with MCP
Certainly
server.
not.
Yeah.
I don't think anybody's using ChatGPT, but we have a bunch of Gemini
Yeah.
and notebook LLM users.
Yeah.
Yeah, to be honest, I'm not super familiarized with the differences and the 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 potentiality of the different bots. ChatGPT, I do know most, but you're right. The MCP thing, I think, is a limitation. I don't know if it's as open as Claude is for you to be able to add connectors and stuff. So maybe that's a very like
Yeah.
strong point.
Yeah.
Thank you.
Sure.
We have a question in the chat from Mike Livestone. Have you done a side-by-side comparison of Claude versus your favorite
Mhm.
non-AI option like NLP, NER?
Yeah, we have not done that yet. And I think before we, publish anything, we probably should do that. And there's some tasks that we can do that we don't need the MCP server for. So that would be a fair comparison, is like taking tasks like that. Um, yeah.
Yeah, good point. Let's see whether there are any other questions in the chat. Okay, we'll just wait a couple of minutes. Meanwhile, I had a question. So I think maybe you touched upon this, but did you expose your existing pathways data to Claude, and did that help in looking for holes or like because no one has the resources to go and
Yeah.
update stuff with
Hmm.
all the staffing shortages and stuff. So
Yeah,
that helps.
that is a good question, and it is one of the use cases that we're planning to try is in addition to new pathway content, update existing pathway content. Yeah. And I think that's where it really helps to have access to the MCP server because it can really look carefully at what already exists. And as soon as it's proposing reactions, oh, do I already have this reaction or do I have these entities? So you're not creating duplications and it's actually remarkably good at that. Although I do think that we have to take a look at how it's querying. I don't think it has this like universal knowledge of the entire Reactome knowledge graph at its disposal when it's looking for connections. But yeah, we should definitely do some more testing, and then we
But
definitely
it could
will.
acquire that,
Yeah,
right?
yeah,
Pretty soon. Like, yeah,
yes, yes, yes.
great.
Sorry, may I add something else? I don't know if we have the time.
Yeah,
Uh,
sure.
oh, sorry. I was just wondering, one thing that I maybe it wasn't clear to me, or I'm not super sure. You were providing the publications, right? The literature. You were not.
No, in the first
Ah,
case, I did not. I did not provide anything. I only said I'm interested in TIM3 pathway. Please create annotations using these resources as references on how to do that. But we have done some tests where um, we we created a prompt where the curator can choose whether or not they want to provide references or have it go find its own references. And that will focus, on the papers that are suggested versus, yeah, going and doing its own
because
research.
also accessibility for Claude for publications that are not open access
Mhm.
is a bit tricky, isn't it?
Yes, and it does point that out. It points out where it doesn't have access. I sometimes wonder, honestly, if it has access to the paper to create somehow to create the annotation, but then it's like, well, curators have to go, this is behind a paywall. So it's almost like it must have maybe covertly gotten the information, yet it can't spit it back to you because it doesn't have, it's behind a paywall. So yeah,
Mm.
but it definitely it clarifies where I mean, I guess from abstracts at least, it's finding that there's potentially useful information and it points out that exactly where they have to go to look for that.
So it's also serving as a triage in a way,
Yes.
right?
Yeah. Yeah.
Awesome.
Great. Well, let's maybe move on right now. Remember, we have like a dedicated time at the end of the three talks to have like an open discussion. Thanks, Lisa.
Sure.
So our second speaker, we're very excited to have her, is Olivia Haley. She is a postdoctoral fellow. Let me pull up her. Olivia Haley is a postdoctoral research fellow at the Oakridge Institute of Science and Education, and she's with the Maize Genetics and Genomics Database, MaizeGDB, where she develops machine learning and computational biology workflows for protein functional annotation and, of course, AI-ready genomics in grain crops. Her work focuses on leveraging large language models and structure-based tools to reduce the manual curation burden at genome scale. And she's a lead author on the journal article detailing MaizeGDB's initiative to define and build a AI-ready infrastructure. So welcome, Olivia, and take it away. You are muted.
Sorry about that. Um, let's see. Can you see my presentation?
Yes.
Awesome. Perfect. So yeah, thank you everyone. And thank you for that awesome introduction. I'm really excited to be here. Um, and yeah, so like it was mentioned, my name is Olivia Haley. I'm an ORISE postdoc working with MaizeGDB. And so recently I've been assisting in MaizeGDB's AI Ready initiative, which in practice not only means that we have data that adheres to FAIR principles, but also that the data can be easily integrated into AI or machine learning workflows and pipelines. And this data is important because we can use it as a basis and a standard to train our new tool, train and evaluate our new tools for biocuration, which is kind of where the link is to this group. So for today's talk, I'll be presenting a short primer on MaizeGDB, um, going over criteria that guided our AI readiness framework, and going over our new data sets that supported this initiative, as well as the tools that we were able to develop using these data sets. And so first, the Maize Genetics and Genomics Database, or MaizeGDB, is a community-oriented USDA-funded informatics service that is focused on the crop plant and model species Zea maize, or maize. So we have a rich history in the maize community, and we host highly curated data, which dates back to before the database was even established in 2003. So we also provide valuable informatics services, and we also actively build tools to visualize and analyze maize omics data. And so this screen is on the right is just a snapshot of the data that we are currently actively managing and building tools for. And so all these tools are managed by a team of 4 permanent USDA employees, led by Carson Andorf, as well as 3 postdocs. But like many agricultural databases, one of the challenges that we've been facing is just the explosion of AI in the informatics space. So, for example, thanks to AlphaFold 2, we now have immense repertoires of protein structures for functional inference. And there's also a plethora of large language models that have come out to assist with biocuration. And we also make space for specialized language models, such as your protein language models and your DNA language models, that have been able to give us insight into functional variants, identifying genomic elements, and identifying function. So, and many of these advancements have come within the last 5 or 6 years, which just demonstrates just how rapidly the AI landscape in genomics is developing. And so, with this rapid pace of AI development, our goal is to make sure that MaizeGDB is strategically positioned to be at the forefront of development in AI for plant genomics, or in other words, that we are AI ready. And so, first, it's important to establish our working vision of AI readiness. And so AI readiness really just adds to the already established and widely accepted FAIR principles. And how they differ is that FAIR principles primarily refer to users being able to easily find, interpret, and reuse data, whereas AI ready is focusing on providing data that enables users to develop AI and machine learning models that learn or generalize biological significance. So, what this means is that data can be fair but not AI ready. So, it can be well documented but noisy and not well structured. Or data can also be AI ready but not fair, meaning that it's gold standard, usable data, but its provenance is not well documented, or it's not publicly accessible. And to this, we identified 3 kind of criteria that really outline our AI ready framework. And the first one tackles file formatting. So, as many of you know, in the genomics field, there are so many different data formats like FASTA, FASTQ, BAM, BigWig, and so many different data types that can be stored within these formats. And while having access to a large amount of data can seem like an advantage to AI, this can be inconsistencies between these formats and structures can make bulk processing really difficult, particularly for those who are more on the analysis and machine learning side and weren't directly involved in its generation. So our first step really was creating structured, standardized, and reproducible data. So that's making sure that the data is stored in a consistent format according to its data type. That we use descriptive column names and controlled vocabularies to limit ambiguity, and that we use formats that support versioning for reproducibility. And so this represents a shift from FAIR principles, where the priority is for the data to be human accessible, to, on top of that, also having data being in a format that prioritizes machine readability. And so our second criteria tackles a major challenge in biological data, which is noise. So biological data is inherently noisy, and there are many different sources of variation. For example, maybe you performed a greenhouse experiment at a different time of year, or maybe for an assay you changed brands. All these can be different sources of variation. And the problem is that because of the power of deep learning techniques to model your variation and your noise, you can actually accidentally start to model your noise rather than true biological variation. So our second criteria involves minimizing bias uh, by marking sources of outliers and noise, by having data sets that have informative features for partitioning into train and test data, and by having data that delineates between experimentally and computationally inferred labels. So while FAIR principles prioritize documenting data provenance, AI readiness adds to this by also prioritizing what methods that reduce feature noise that's seen by machine learning models. And the last criteria addresses making the pipelines and data available for use, which is along the lines of FAIR principles. But what adds to this is making sure that you have a resource to document reproducible data sets in computational pipelines. And then also providing pre-computed data sets that can be machine readable and readily used. And then also providing a resource for large-scale feature extraction to build customized data sets. And so this shifts the focus from not only encourage data sharing and reuse and attribution to encouraging the adoption of machine learning and AI within the community. And so taken together, these are the criteria and sub-criteria that have guided our initiative. And so there were various steps that we used to sort of check off each of these boxes. For example, we standardized how we stored our data by committing to using specific file formats. And then we also made our pipelines and workflows publicly available through GitHub to foster community adoption of AI. But for today's presentation, I'd like to take a deeper dive into the AI-ready datasets that we curated to support this initiative. So in 2024, Anaconda launched a survey asking over 1,000 data scientists, how do they spend their time with data? And their respondents answered that on average, more than a third of their time was simply taken up by data wrangling, so data preparation and data cleansing. And so fields like computer vision and computational chemistry have been able to minimize this time or this data wrangling stage by using standardized data sets. And these pre-cleaned, predefined data sets not only save time on data preparation and cleaning, but they also accelerate hypothesis testing and serve as a standard or a baseline for model evaluation. And so we saw that there was a need for AI-ready data sets in Maze. And now I'm going to walk you through a couple of these data sets that we generated for our initiative and some use cases. Here we go. So a great way to demonstrate these points is starts with our pre-computed protein and DNA variant scores. So these zero-shot scores are computed by protein and DNA language models, which are trained to predict the identity of masked or missing base pairs or amino acids. And so this allows them to learn sequence conservation patterns and essentially learn when it's okay to substitute different base pairs or different amino acids versus what substitutions are not naturally found within a population and therefore may be consequential. So the learned probabilities of these various substitutions are then used to calculate log likelihood scores or zero shot scores that correspond to their potential impact on the DNA function or the protein function. And these and more negative values indicate that the substitution may be more deleterious than more positive or than less negative scores. And so these models produce powerful metrics to predict functional consequences of allelic variants. And we've actually integrated these scores into various tools. So, for example, this is a snapshot of our SNPversity tool, which surveys 250 million single nucleotide polymorphism sites across 3,000 maze accessions. And for the SNPs that result in a point mutation, it shows the variant effect scores that are calculated for the DNA, as predicted by plant CAD scores, and the protein, which we used ESM variant scores to show. And I also wanted to give you just a working example of how this tool, which integrates DNA and protein variant scoring, can be used to accelerate plant breeding. So this slide focuses on the gene sugary 1, which encodes a starch debranching enzyme that is defective in different varieties of sweet corn. So this mutation causes a higher sugar content within the kernel, but then also a characteristic wrinkled phenotype. And these images are just showing the different the different phenotypes for the different mutations in this protein. And so the table to the upper right shows a summary of the SNPversity results for this gene, which include the wild type or the reference base pair, the allele variant that's found within the population, the resulting point mutation, and then the predicted effects to the DNA and the eventual protein. And what's really cool is that these scores reflect experimental data that we have on these point mutation mutants. So, for example, these variants that are highlighted by the red box were naturally seen in the protein's function or in the population, but the literature suggests that these don't have a major impact on the function of the protein. And so accordingly, we see that these tend to have a higher variant score than the other mutations, which actually cause a major change in function. And we've also integrated these variant scores, particularly the DNA variant scores, onto our JBrowse tracks, which show a close alignment with the peaks show a close alignment between the coding regions of the sugary 1 protein, but then also other genomic elements like transposable elements. And so this just shows how these DNA and protein language models can detect a wide range of structural elements in the genome and their impact on protein function. And so we also developed a feature vector of the top 60 features, which includes data that spans across gene structures, tissue, mRNA, and expression features, protein structures, and biochemistry features. And so some of this data is available for our reference B73 genome, but much of this data is also available for our non-reference genomes, which makes these vectors just really powerful for predicting or for developing models for our reference, but also non-reference genomes. And these data sets are also useful beyond machine learning purposes because the features within these data sets can be also used for doing descriptive statistics on gene sets or even functional enrichment, which is what I'm going to show in the next slide. And so one of the ways that we use the core 60 vectors was to develop a platform at MaizeGDB for functional enrichment term analysis, which we called FETA. And so functional enrichment is among one of the most widely used methods in bioinformatics. But for Maize, many of the existing platforms limited analysis to solely the single reference genome. But other genomes are rich reservoirs for disease resistance and other important agronomic traits. And another problem with other platforms is that they usually only allow for enrichment in one or two ontologies. So, like Go ontology or pathway enrichment. So we designed Feta to allow users to perform multiple simultaneous enrichments of Go terms, pathways, enzyme functions, and many other sources of variation within our reference B73 genome, and then also our non B73 or non-reference genomes. And the platform also features other analyses that can be used for analyzing gene sets on the basis of evolutionary conservation. And we also recently integrated RNA-seq data into these analyses, and we've integrated a way that allows users to upload the results of their RNA-seq data from popular platform or popular pipelines like DE-seq and EdgeR. And using Feta, they can upload this data, visualize the upregulated or downregulated genes, and then directly send them to either of our two functional enrichment pages. And so I just wanted to give you a demonstration of how Feta can be used to devise trends in functional analyses. So this data set comes from Shurak et al., who compared the transcriptomic response of a corn smut resistant line, which is called CML322, and a susceptible line, which is our B73 reference. So we took their 400 upregulated genes within our resistant line, and we ran simultaneous GoTerm, enzyme commission, PFAM domain, and subcellular localization enrichments to identify interesting trends. And this CNET plot just shows the connectivity of overrepresented protein family or PFAM domains, which is interesting because they pointed to an overrepresentation of membrane-bound proteins with NB-ARC domains and protein kinase domains, which are the hallmark proteomic or protein architecture for disease-resistant genes called NLRs. And so four of these proteins that are highlighted here, that have this architecture actually actually mapped back to a locus that was previously identified to be involved in resistance to Puccinia sorgi. And then two more of these were also identified in the literature as potential resistance genes to Puccinia polyspora or southern rust. And one of these genes was also identified as a candidate resistance gene or candidate resistance protein, RPM1. So this example just demonstrates our AI-ready data that we're able to use to build an application that helps our users take their gene lists and within seconds surface some sort of mechanistic hypothesis. And then from an AI readiness perspective, FETA could also be used to characterize genes that AI models consistently get wrong or mischaracterize, which is also a powerful example. So the last example or the last data set that I wanted to highlight was our curated literature data set. So MaizeGDB has a long history of manual curations, and some of our curations span back to the 1920s, which is actually where this image is from. And so we assembled these into a data set. Or we assembled these into a data set that could be used to train and evaluate the efficacy of LLMs for LLMs or just any other machine learning pipeline for biocuration. And so this data is actually being used by one of our pipelines to evaluate the use of a pipeline called Texpresso, which takes PDF documents, tokenizes them, performs various analyses, and then integrates or performs various analyses to extract annotations, and then integrates them into a web interface. And so this protocol can save biocurators time on extracting annotations like gene models and traits, methodologies, and gene function. And so this is the publication where I have presented much of the data for this talk. Feel free to check it out, and if you're not able to access it, feel free to reach out to either myself or Carson Andorf, our corresponding author. And with that, I would like to just thank again the MaizeGDB team. I'm so fortunate to be a part of this team and this initiative, and have access to all of their knowledge and support. And now I'll take any questions that I have, provided we have time. Thank you, everyone.
Thank you, Olivia. Yeah, we have a couple of minutes. Anybody has questions? There's more, I guess, of a comment in the chat. Mike says that that number, I think this refers to the time spent on sort of data preparation, data cleansing, which was sort of your one of your early slides, that number can be high as 50 to 80% in some studies.
Yeah, yeah, that's true. And yeah, I've definitely seen that in the literature as well.
Yeah,
It's a
any
great
other
point.
questions? People can either raise their hand or type in the chat. Yeah, Olivia, I really liked your point about having the intersection between fair and AI ready, because I haven't heard too much about that in all the AI talks I've been to. So I guess one can help the other, right? That having data sets both fair and AI ready. So that was a nice point. Another quick question I had was how much of the whole vocabulary and about data sets, how much is that standardized across the plant community? Because one thing that we did a lot in Alliance of Genome Resources, I don't know whether you know too much about the Alliance of Genome Resources, but it's a portal for, most of the major model organisms. And we spent a huge amount of time just standardizing and, across data sets, even just the vocabulary that different databases use, say fly versus worm versus mouse. So how much of that needs to happen before you can even expose or train AI models? And has the plant community done that?
Yeah, that's a great question. Which also speaks to just the difficulty of standardization, because we do share some standardized or some standardized ontologies like the Go ontology and KEGG as well.
Yeah.
But yeah, there's so much variation between from plant to plant that it's really difficult to come up with a standard ontology that's going to fit every single use case. And we've sort of tackled that by using just different ontologies that people can sort of pick, in between, because every ontology is a little different. Um, and I'm not necessarily sure that beyond having like Go terms and keg, that our ontologies need to be super specific or need to be standardized across plant species, just because there's just so much variation and so many different crops that have sort of a different pathways and different agronomic traits that people care about, sometimes you really need a more crop specific viewpoint rather than something more generalized. But I could also others can also argue the opposites.
Yeah,
So
I think good point. Like a balance of kind of crop specific and then generalizable, I guess would a good mix would help, I guess, the model learn better. I don't know.
yeah.
Yeah. I think Lisa has a question, has her hand up.
Yeah, I just was curious about your use of Textpresso and the type of annotations you're extracting that way, and is it fully automated, or is you the curator in the loop to sort of check what's going on, and how does that work?
Yeah, so we're still in the kind of early stages of setting up Texpresso. I think the postdoc who's working on it just got it running. So we're really trying to see. We're really at the stage where we look at, what what can it extract, and kind of put it through a stress test and compare it against extraction using like an actual LLM, as Texpresso is more machine learning based, rather than incorporating natural language processing. Um, so we're still at the very early stages of this project, but I'm looking forward to its results. We're also collaborating with the with Ag Biodata and Kapil Chugle at WormBase to also test it between not only with maize data but also with sorghum data. So we're going to have a very robust comparisons that are between species and also between platform. So stay tuned, I guess, is the
Okay, thank
is
you.
Yeah, yeah,
thanks.
I got really excited when you mentioned Texpresso because Texpresso was originally developed at WormBase, and I started out my biocuration career in WormBase. So I will go back and tell Michael and the team that, hey, Maze GDBs, using Texpresso.
Yay!
Yeah. Hans Michael Mueller, actually, we worked in the same room, and that was when he was developing Texpresso. Yeah.
What a small world.
Yeah, I know. Yeah. There's one quick comment in the chat. Mike says bespoke ontologies are fine until you need to combine the data with someone else's. Yes. Then that becomes, quite the pain. Yeah.
Yep. And that
Yeah.
definitely speaks to your point of balancing the generalized versus more crop specific.
Yeah. Yeah. Okay, great talk, Olivia. Thank you for stepping in quite at the last moment because I think Dr. Andorf had to travel. So thank you. And we're excited to have, more younger members like grad students and postdocs present because I think it's good exposure for them. So thank
Yeah, thanks so
you.
much.
Yeah. So let's move on to our last talk. Chris Tabone is a colleague of mine at the Alliance of Genome Resources. He's a senior programmer analyst at the Jackson Lab in the lab of Carol Bolt. His work centers on database architecture, systems management, and the development of biocuration software for Flybase and the Alliance. He previously held postdoctoral fellowships at Trinity College Dublin, where he studied fruit fly behavior, and Harvard University, where he focused on neuronal modeling. Prior to his current position, he served as a biocurator for Flybase. His expertise lies in the software integration and infrastructure of model organism databases, with a recent focus on the application of large language models to enhance biocuration workflows and tool development. So Chris, take it away. So we're also doing something special here that we invited Chris back because when he first talked about the his tool, it was being developed and it had reached a certain stage, and now there's a lot more exciting things that have happened. So we're happy to have him back to sort of continue the story of his tool.
Great. Thank you very much for the introduction. Um, Hello, everyone. I'll share my screen in a second. We have a fairly intense thunderstorm rolling by. So if I drop off the call, I apologize. I
Oh
haven't lost power
no.
yet. Yeah, I have backup for my computer, so hopefully I can keep talking. But I just wanted to point that out. That's why I would leave if it happens. I hope not. Okay, let me share my screen here. Okay, track of my time. So, as Rajata mentioned, this is a kind of a part two of the talk. I spoke back in, I believe it was February, and talked a bit about this system that we're building at the Alliance of Genome Resources for literature curation. And it's been a few months. We've made some progress. I will show a couple slides here and then jump into a live demo to show the website as it's working now. Very open and would love feedback about the process that we're taking, or if people have any questions or comments. It's very much appreciated. And this is a system it's still pre-release, hoping maybe to get something out by the end of the year in a more polished form. And it's being built for the Alliance, but also being built in a way that can be distributed and used by other organizations. So hopefully once we reach version 1.0, if other people are interested in adopting this technology, they're more than welcome to do so. And we've been developing it with that in mind. Okay, so a brief overview here. This whole thing started with just a simple question that I know many people here have asked, and I said this during my last talk too, which is how can we use these LLMs for curation at the Alliance? And we've heard some really great answers to that question today, and what we're coming up with here with this software is not entirely novel. There's been many groups that have been trying this approach. We made some decisions and we have some frameworks in place that we feel works best for our environment and our data structures and our situation here with curation, and hopefully that other groups find it useful as well. So I want to just spend a minute with two slides to talk about the overall design of the project and the agent architecture that we're using. I'm going to talk about the changes from the version I first showed a few months ago to the version that we're on now. And then I want to dive in and spend a little more time on our evidence-first extraction and an AI agent development workflow that we use to quickly work on this project. So that's outside the actual curation. It's more on the development side of things, but I think it's interesting and maybe useful for people that are looking to create tools that are similar to this. This is the very generalized and high-level view of our system. The BioCurator assistant in the green box there, that is our UI interface, and that's kind of the heart of this program. We use agents, so we have individual agents that are running. They're backed by OpenAI's LLM at the moment, GPT class LLM. We can use ones from Gemini and from Anthropic if we need to. We do actually have an Anthropic model in there too, but it's more for conversation with curators to help them develop. It doesn't actually run the agentic code that we write. And we are processing PDFs into a vector database. So we create these things called embeddings, which is basically extracting the data from the PDF and putting it into math. And then the agents can query this and find information following the curator's instructions. So it looks for things that the curators ask it for in papers. It can output it in a variety of formats, and it checks everything against local databases that we have and external databases too. And importantly, as other people have mentioned in their projects, it's the same with this project, is that the curator is in the loop and they're the drivers in this program, and nothing is entered or used in this program without their explicit verification and approval. So that's really important to our design methodology. For the agentic architecture itself, we have a supervisor style model. There are many ways you can structure this with AI agents. The one that we're going with right now is that we have one supervisor agent that runs a very strong model, and it can dispatch other agents depending on the task at hand. So a PDF extraction agent or an agent to validate genes or an agent to produce output. Those agents, depending on the task, can be weaker models to save cost, time, and tokens so they can work quickly if they have non-complicated tasks. Or they can also be strong models like the supervisor agent if they're doing things like extracting data from papers, where you need a really strong, powerful model to do a good job in that sense. So it's an agent dispatching other agents. And this can be done dynamically through our chat interface, or curators can define what we call flows, where they explicitly run a series of agents in a particular order to do reproducible extraction from PDFs. So we have two ways really to interact with our program. One, again, is just chat, and the other one is creating more structured pathways for the program to use and follow. Okay, so just to go back to this again, as I mentioned earlier, the first version was our 0.1 version. We're on to 0.6 now. When we finally do release to the public, we'll increment that first number. The four things I'm going to show after this slide, the first two I mentioned earlier, so the evidence first extraction, I want to show kind of the workflow that we have that runs underneath our whole program. And then I want to talk a little bit about the rapid development to actually make the program. And then the two last things I'll show on a live demo. So I'll show how curators can create their own agents using the user interface in the program itself, so on the web UI. And then I'll show the very first early version of our curation interface and how basically you go from questions to a paper to actually getting it into a format where it can be entered into a database with a couple clicks. Okay, so this is a lot here to throw up on a slide, but it is the general overview of the whole pipeline that we use for this program. There's a lot of iteration that went into this. There's a lot of trial and error that went into this. It's people might have heard the term a RAG pipeline. It's very similar to that. It uses a lot of the aspects of that. So it's basically our own version of a RAG pipeline with an AI in the mix to do a lot of the querying. So I'll walk through it real quick. The very top part is all our paper processing. So we have PDFs, we extract the data from them. We have a standalone program that's also distributed with this curation program, as we call it PDFX, which is the PDF extractor program that pulls data out of PDFs and then it chunks it into sections. So we chunk by sections of the paper. So chunks are just kind of like a gathering of data that represents the information from a paper. So we have things like the introduction would be its own series of chunks. The discussion would be its own series of chunks. So you just kind of pull the data out of the paper, you organize it, and then you can query it with math. So we pull out the data from the paper, we embed it, we chunk it, and we use a database called Weaviate. There's many versions of these vector databases that are used for this kind of information processing. Weaviate is just one that we chose. It works very well. When we put the paper into the database, it does turn into numbers in a Euclidean space with this embedding stuff. It's all very, very mathy, like I mentioned earlier. But we also preserve the sections of the paper. So when the AI agent in the second row goes to do searches into the database, it knows the labels and the titles of the section. So it knows, oh, I can look in the introduction section of the paper. I can look in the results section of the paper, or the experiments section of the paper, or the discussions part of the paper. So we leave those keywords, we capture those keywords, so that when curators come to ask questions, the AI can search for words. This is this hybrid search here. So it can say, if you ask it, what genes are the focus of this paper? Well, yeah, it can go look for genes in the paper, but it also might make calls say, okay, well, I better look in the introduction first, because that's usually where the important parts of the paper are outlined, and I might find what genes are the focus just by reading the introduction. And you'll see the actual agent do that as it does searches. So we allow it to search throughout the entire paper, but we also retain, like I said, the sections of the paper as part of the knowledge in this database. So the second part is all the real nerdy brains of the program here. So the AI will go in, it will do different searches against this database, it will pull out relevant chunks, it will read sections or subsections of the paper that it finds are important and necessary for this, and then it produces a candidate record. So it says, these are the things I think are important from this paper, and it brings them back to the curator. And the bottom part is arguably, I think is one of the most important parts, which is that the entire flow is heavily rooted in evidence. So when it produces these candidate records, we require it to present quotes directly from the paper, and we also verify those quotes. So if it says that, and the example I'll use, the paper is has a gene called crumbs. It's a fly paper. So if it says that crumbs is important in the paper, then we need to see sentences that support that decision. And the LLM pulls those out of the paper. We verify them programmatically. So for the verification, we're doing this with Python scripts and stuff like that, just to make sure that nothing is hallucinated and that these evidence quotes are actually rooted in the publication itself. We package this all up programmatically into what we call an evidence envelope, and then that gets reviewed by the curator, complete with highlighting. And so they say, this is the claim that the LLM is making. This is the evidence in the paper. There it is, highlighted right there. Do you want to accept it or reject it, or should we go back and revisit this? Is it missing stuff? So we try to keep it extremely clear to the curators what's going on. We have an all audit system and history system, but then also, again, really rooted in the evidence. And addressing hallucination is probably one of the most important things we do in this program, because it's definitely a problem if you go into this to get trustworthy results using a lot of these tools. So moving on to the development of the actual system itself, we're using something called Symphony, which is a program and a framework, I guess you could call it, developed by OpenAI. It uses what they term harness engineering, which people might hear more and more as it becomes more common. But it's a way of using AI in a very autonomous way to do very well-defined tasks very quickly. So what we do is we take curator feedback from this program. We have many ways for curators to give feedback to us. We have a program called Trace Review that also ships with this program. So Trace Review is a program that reads all the LLM calls that were made, all the tools that were invoked, all the things that the curator asked and the LLM responded with, the logs, the database entries, all of it in a very quick manner, and returns all of the data back to an LLM to review. So when we're developing the program, we're also using AI agents, and we're doing so in a way that's very thorough and very robust and very efficient. So a curator feedback comes in, we run this trace review investigation, and they might have a feature request or a bug or something like that. We can quickly create tickets for that. We use Jira and another app called Linear, and then we can dispatch this to our Symphony agent, which are powered by GPTs, and actually work autonomously to create the code that we use in this program, which then gets gated by a very large number of tests. So we have automated testing gates that need to pass, and then we also have human review of the code before we ship it and can consider it complete. And this rapid development lets us do things like, I think it was just yesterday or 2 days ago, we released a new version of this tool internally, and there were a couple hot fixes that had to go in, and Sue, who's on this call, was working on it, and she couldn't save her custom agents. And that kind of debugging would have taken probably a few hours for a person, but we were able to push a fix in about 10 minutes, 15 minutes using this pipeline here. So Sue submitted curator feedback. We ran the investigation, dispatched an agent to fix it, reviewed it, and pushed it all within about 10 to 15 minutes. So we've been able to iterate really quickly on the development of this tool using this approach. And if people are looking to do AI development, I heavily recommend looking at Symphony and some of the newer OpenAI agents, because they're very powerful for this task. Demo. Okay, demo time. So if people can still see my screen, I will switch to the curation tool. Is this showing up for everybody? Can people see it? I'm just going to take a sip of tea right here.
Yep.
You can see it.
Yep.
Okay, awesome. So some people have seen this. So Sue and Ranjana and a few others that are on the call, Ryan Taylor, have seen this tool. I want to show a couple things. First, I'm going to go into the Agent Studio and show how curators can make their own agents. And this is new behavior. This is different from what I showed in February. This is kind of an extension of the program, as it were. So this is getting a little crowded on top. I'm probably going to condense this into categories soon. But Agent Studio is a way where curators can go into here and interact with the program in various ways. Whether it's learning about how the agents work by clicking on this and reading descriptions about what the agent's jobs are and what they do, or creating the flows that I mentioned earlier. But I want to look at the agent workshop. So the agent workshop I should mention quickly. So this UI is a little poor at the moment. This is kind of a first iteration of this, and we'll polish it off a little later. I'm kind of developing with the approach of getting the tools done and into the hands of curators, and then we can iterate on UI when there's more time. But if curators go into this agent workshop, they can actually create their own agent to use in this program on their own. So you can start from scratch. You can clone an existing agent that you might have made or use a template agent. So here's a whole bunch of list of agents here that have different tasks that they do from gene extraction, to chemical extraction, validation agents that interact with the database. So if we pick a specific agent, you can type a name here for the agent and pick an icon to use, type a description of the agent, and then you can pick the model that it uses, whether you want to share it with other people in this project. And then you can learn a bit about, if I change to a different model, what's the pros and cons, avoid for this, recommended for this. So curators can pick models and reasoning levels that they think are appropriate. And there's some kind of guidance here for them to use. What kind of models do you want to use? And the nice thing about this is you can iterate. So you can create your own custom agent here, pick a model, pick a reasoning level, try it. If it doesn't extract things the way that you want, you can come back here and fix it up more, change it. And when they scroll down to here, there's a main prompt that they can modify. So this is what the agent is going to use in order to do its task. And then we have what we call group prompt overrides. So if you're a Flybase curator, MGI curator, RGD curator, you can put specific rules in for your mod. So if you want your agent to behave in a way and follow curation instructions that are just specific for yours, you can enter all of that here. And then once you make all of this stuff and you make your changes, there's tools that you can see here, and you can read about what tools you want attached to this, and you can even request a new tool and send a request to the developers. The tools are written in Python code, and these are ways that the agents interact with things like internal and external databases and things of that nature. So there's a preset number of tools here, and some of them like record evidence you need to have, but other ones like searching documents or doing a curation query against the database, those are ones that developers can help curators make. So if you say, I want to make an agent, I want to go talk to something in EBI. Oh, I need a tool for that. So then we'll have a conversation with you. We'll create this tool that will go talk to a database that's hosted over at EBI, and then that'll be available to the agent that you're working on. So tools still require some developer intervention because they're kind of complicated Python scripts. But you can, again, if you're a curator, you can request it. You can talk to Claude about what your tool might look like. And then you can put that request into to a developer and they can help you with it. And then after you've come up with your tool here, you can save your agent and then you can use it in flows. So then you can load your agent into this flow here and then use it in the program to analyze papers. And you can iterate on this process. And we've had some curators work on this for several iterations to help improve these prompts. And this is kind of an important point I want to mention is that I'm coming at this, I have a lot of biology experience. I was a curator myself for a year, but my prompt knowledge would be heavily into fly literature. I don't have the ideal prompt making skills for making things for rat or for mouse or something like that. So this kind of tool lets a curator from one of those specific mods come in, adjust the prompts following their curation guides, and then actually test it in the program to see how it performs. And we have Doug from Zephyr, Doug Howe is working on this, and he's been iterating on many prompts to kind of optimize the zebrafish curator prompts to work well in this current system. And then you can also at any point discuss this with Claude. So if you click this Discuss with Claude button, there's actually an agent that runs in the left panel, and you can have a conversation with this and talk about like, oh, does this prompt look good? Do you have any recommendations for it? It has risks that it might tell you or ambiguous instructions like, what does this mean? Confidence must reflect the reliability, and it'll give you suggestions. And a lot of these suggestions are rooted in how the program works. And so this agent that you can talk to on the left side can help steer your development of your custom agents. And it gives some validation tests. It's being very generous with the recommendations here. So as we develop the program, we also update the knowledge of this Claude chat agent here. So if I make changes in how the program works, I make sure to say, okay, I got to update Claude in case the curators speak with it so that it understands, what's ideal now, what are the new tools that exist, or what are things that it should push curators to design in these prompts to make them better or worse. And you can always send messages with this conversation back to developers. So you can say, I talked to Claude here on a custom agent for a long time, and it had some good stuff, it had some bad stuff. If you click this button here, it'll actually send me an email with the conversation, and we can take a closer look at like how the conversation went, what tools came out of this, what changes came out of this. Are there changes in like the base prompts we have to make for all of the agents, things like that. So a lot of it is trying to be very transparent, very interactive between the curators and development to help this process go quicker and smoother. So I'm going to hop back here to the main screen. And I'm going to ask a quick question here because I want to show in the few minutes, I have 2 minutes left, to show how the evidence works and how the curation flow works. And this is an earlier version of this. So again, the UI is a little rough, but the general principles are sound and it should work. So if we ask something like, what are the alleles in this publication? Well, that's a simple question here. On the right side, you can immediately see the audit logs start filling up. So that's kind of a transparent window into what's happening behind the scenes. And you can see all the tools that are called, all the agents that are called, which ones are being called, what they're being asked to do, all again on the right column. And as they start actually digging into the paper, which they're doing now, you'll see these record evidence flags come up. And that means that it found something and it successfully recorded the evidence for it. And you can even see it here. It's querying the curation database too to validate the validity of some of the alleles that it extracted from the paper. When it's done with all of that, it'll do some reasoning. It'll send the data back to the supervisor, and the supervisor will return the output to the user in this main chat window here. So I asked it, what alleles are studied in this paper. It found 3. These are correct. These are all the crumb specific alleles. And if I were to go here and I click on these evidence quotes, these little chips at the bottom here, so we'll just pick this first one, and then I further click on these, it highlights the section in the paper that matches this. So it says these 3 alleles are what I consider the important ones from this paper. And then here's the passages in the paper that I find the most important to deal with those alleles. And there's no limitation to how many chips can be presented for each piece of evidence. Sometimes it will bring back multiple chips. So this CREB 8F105, if it thinks there's 3 passages that are important, it'll say 3, and there'll be 3 of these to click through. And you can see another example here for another one. So the whole system is quick and reliable verification of what the LLM is finding in a very transparent manner. And you can see this one as a good example here because it says 2 of these, blah, 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 and talks about these alleles. And this is the first time in the paper it mentions this allele. And it considers this introductory passage very important. So after you do this initial query here, you can do prepare for curation. We'll click here and you hit start prep. Now this brings you to a curation screen. Again, this is an early version, so the UI is a little rough. But it's these same 3 alleles now. The evidence is on the bottom. It has the same 3 click-through thing here. And you would be able to accept or reject these things as being, proper answers to the question that you ask. So if you want to tag these entities as existing in the paper, you can quickly read on the left here, zoom in a little bit if you need to, read the passage. Yeah, that looks good. Hit accept or reject it. And eventually we'll be able to have a button here to resend the LLM off with some additional instructions. Like, oh, you were close on this, but there's 2 other things that you missed. I want you to go back and look at that again. And then this will update in real time. And then the way that we export this, we're basically trying to standardize it, will be this assembled payload with data. And you can see these are blocked because we didn't accept or reject them. But if we go here and we accept, we'll just quickly accept these 3. We look at what our submission looks like. This gets populated and gets sent off to our actual curation databases or exported to TSVs or CSVs. You can download these bundles. And again, in the mindset of keeping this open source and available to everybody, this export thing can be modified to suit people's databases, or we can make a standard JSON format that anyone could use any of their tools to then incorporate this and ingest it into their database. So we're trying to keep this model output at the end very standardized as much as possible. So yeah, it's a work in progress. We've made a lot of advancements in the last few months, and hopefully we will have a system in place by I'm hoping the end of the summer to actually be incorporating some of this into a live database. There's still a lot of work to be done, trial and error. And Sue, who I mentioned earlier on the call, just the other day was talking about strains. We have to refine we were missing a lot of agents here, probably going to get up to at least 100, I would imagine. And we need to work on the prompts and connect more data to these agents. But it's promising and it seems to be reliable. And so far, it's a very useful tool. So I'm going to pause there. I went to 23 minutes, so sorry. But yeah, I guess I'll stop there.
Thank you, Chris. Great talk. So we'll take any questions.
I think that's just a automated note in the.
Oh, okay. Yeah. If anybody has any questions.
Just a comment. I'm blown away, honestly. I'm so glad your talk went after mine.
I loved yours, Lisa. I thought it was excellent. It was, I mean,
So
so like, okay, to that end, I just want to say, like, we started with a lot of what you're doing, and the way you're doing it is still very powerful. And the way you're doing it doesn't rely on like a super nerdy dev, as I push up my glasses, working on this 24/7, So there's a lot to be said about your approach, and I really like the way that Chris Mungle champions it because I think it makes this very approachable to people with no programmatic background. And I think what we're building here is just the next step. So it's like a bit more harness in line to like, are we verifying the publications, verifying the identifiers, verifying, you know what I mean? So it's kind of like what you're doing, but just with more
Well,
more
yeah,
guidance.
it's automating. I love the way that you've somehow already had it check itself with the excerpts that it's checking before it even sends it to the curator. Because
Yeah, and there's a
pull that
little
out,
loop built in
but
there
you've done
too.
it automatically. So that's just
Yeah, there's a
yeah,
loop there too where we send it right back to the LLM. If, we'll
yeah,
say, you said
yeah,
something, this is
yeah,
fabricated, go back.
yeah.
And if it can't find it, we reject it. And then,
Yeah.
that's
And it's amazing because it's exactly the same thought process, but it's all
Mhm.
manual and you've done it like automatically. And that's totally what we're
Yeah.
missing.
Yeah.
It's just very impressive. And I'm so excited about the idea of being able to maybe kind of interact with that at some point and for our kind of goals. So really looking forward to seeing where that goes.
Yeah, we've had so Seth Carbon from Go has actually um, deployed it at Go as a test to see if another organization could get it working, and he was able to. It took about a day. So as we develop it, it's Docker based, it's portable, and I'm trying to spend a lot of time into making it so that other groups could just deploy it at their organization if they want
So
to.
you'd be willing to work with us on
Oh,
trying.
absolutely. No,
Oh, that would
absolutely.
be so
When this goes
amazing.
live, I think it's a valuable contribution to the
Yeah.
community. I'm really enthusiastic about that. So when we get a public release and we publish it and whatever, like, yeah, we'll have support for this, provided we have the time. But I very much want to make that part of this.
Right. And I love the way that you can show the article, like you can actually pull up the article automatically. That's amazing. And I'm just curious about paywall articles. This is all
Yeah.
yeah, PubMed
So
Central.
yeah, that's a so right now the curator can upload whatever they want, and um we have to so yeah there's a murky ground of if you take something that's paywalled and you chunk it and you embed it in a database, the part where it searches the data, it is sending chunks and data to Anthropic servers or OpenAI servers with bits and pieces of the paper. So there's a legal gray area there where it's like, okay, well, this is a paper that we have a license to have. Is it
Mhm.
okay when we're searching mathematically into embedded sections to like send that data off to OpenAI's server? I mean, if it's not okay, then that's a problem. If then we would be limited to open source papers. But yeah, that's something we have to talk about and discuss. And the things we're using now are mostly, if not all, I think open source papers too.
Yeah.
But yeah, that's a I mean, we're all doing that. If you're using
Yeah.
Claude Pro, we're all doing those kind of things. So hopefully that's acceptable.
Yeah.
Sue has had her hand up.
Yeah. Hi, Sue.
Yeah, yeah. Hi. I figured I would chime in here. I sort of had one question that has been nagging at me as we go to all of the meetings for the Alliance, which is, we talked about it a fair amount there, but I didn't hear too much about it other than, guarding against hallucinations, but just making sure that people think about what are the guardrails you need to put in place? How do you keep Claude or whatever AI you're using on the rails? And just making sure that we're thinking about it and what are people doing to think about that so that it doesn't, go off and do the horror story of deleting your entire database because you told it to make some change and it decided to do whatever it wanted.
Yeah, I mean, so we had a discussion to that extent. I mean, there's if you're running stuff like Claude Pro and you're on the web UI, it's a little safer. The program that I'm using is running on our servers, and we spent a lot of time making read-only profiles for the databases and limiting what it's able to access in terms of APIs and things like that. It's always on the back of my mind that one of the first things I do when I develop agents to be used in this thing is immediately think of the blast radius. If this thing became completely evil, really, this is the question I ask: what could it break? And then I go from there and I develop the guardrails first and foremost. And like the examples I showed, it is querying the production database to verify the identifiers for the alleles, but it's doing so on a restricted version of the database with a read-only user, and it's very, very nailed down. There are issues. There was I think about a month ago, there was what they call a supply chain attack for Gemini, where somebody snuck some bad code into one of the packages that the Gemini LLM uses, and it could do prompt injection. It was only for like 4 hours. It was scary. So that if you ran that on an unprotected server, it would harvest all of your like SSH keys and send them off to some organization. So there, we try to keep these things under wrap as much as possible for reasons like that, because they're kind of like rogue in that sense. You know what I mean? It's very strange working as a developer with something that is not deterministic, where it's just like, I'm going to do this thing because I think I'm helpful. And it's like, what did you do? Don't do that. So yeah, that's at the forefront of all development thought.
Yeah, exciting conversation, but we are over a minute, but that's great conversation. So what we may do is like really have a session in one of these series to just talk and interact maybe and have like open discussions. We could do that, like plan one of those sessions. So I want to thank all the speakers like Lisa, Olivia, and Chris. Thank you so much for coming and giving great talks. And I just had, like, I was told that I need to have closing slides. So here we go. So these are our upcoming events. So first of all, the ISB Early and Advanced Career Award nominations are open. So please nominate yourself or your colleagues for awards. Only members can vote, but non-members can be nominated for career awards. And so the nominations close May 15 and voting is June 1st to the 5th. Details are here. And we have more events in our virtual conference sessions. We have a June 15 virtual poster session. There's a link there, and go to our website and the events and the Biocuration virtual within the events, the Biocuration 22 virtual series. September 14th, we have a careers in biocuration talks, a great panel discussion. And in October, the date is to be announced, we have the ISB annual general meeting. So please become members. Tell your teammates to become members so that ISB can serve you better. So with that,
Yeah.
I'd like
Charlie
to
and I dropped links in the chat for all of the various submissions and upcoming events.
Yeah, thank you. All right, so we'll see you again maybe in another one of these before the end of 2026. Thanks, everyone. Bye.
Thank you.
Bye, thank you.
Thank you. Bye.
Thanks.